her PhD at the University of Sydney in physics in 1994. She started her career initially working on Venus and mostly working on near-infrared astronomy. She ended up coming to the US to work at JPL, where she was the lead research scientist on the Spitzer Science Center, and received many awards from NASA for group achievement for activities she led associated with that telescope. So early in her research in that area, she started thinking about about exoplanets, which could be visualized initially with Spitzer, and certainly you've had many telescopes since then that can visualize them even better. But she's also maintained that connection as moving into exoplanets with Venus by realizing that we have examples of exoplanets just in our backyard, in our own solar system. And much of what we know about anything in astrobiology and anything about potential life in habitable worlds, we know from Earth and other examples in our solar system, which has been one of the major contributions that Vicki has made to pushing forward the area of exoplanets within the astrobiology community and for astrophysics. For the last several, or at least 2 decades, she's been an international and national leader and very well respected and included in all sorts of activities and development of strategies and roadmaps for our program. She's a consultant for a group in Japan, where she guides their research and reviews their research. And she's, as I said, recognized internationally as well as nationally. She served on several National Academy committees. She did double time about 5 years ago on there are 2 panels that were directed, or studies that were directed by Congress to look at astrobiology and the search for life, a strategy for that, as well as looking at the direction for exoplanets and the connection. And she was popping back and forth between these two committees. She also then, of course, the astronomy and astrophysics decadal, which is something that is directed by Congress to do every 10 years to identify research direction and, more importantly, missions that are supported by NASA and potentially NSF. She was one of the leads for exoplanets and astrobiology, one of the science leads, or led that panel. So again, she's played a major role. For me, when I came on, I'm an oceanographer, a biological oceanographer. I've done work in Antarctica, but I didn't know anything about astronomy. And I really relied on people like Vicki, as well as John Barros and several other people that are in the audience, to help me understand the breadth of astrobiology. Let's see. I want to talk a little bit about some of the awards that she's received. She's a fellow of the American Geophysical Union, a fellow of the American Astronomical Society. She was elected to the Washington State Academy of Sciences. She was a Tinsley Visiting Scholar. I mentioned before that she has numerous NASA Group Achievement Awards. I mentioned about her work with Spitzer, but she also has one being part of the Astrophysics Large Mission Study Team that came up with 2 concepts for studying habitable exoplanets and searching for life. I mean, the list goes on. She was also got the Drake Award from the SETI Institute. I mean, again, and actually, this is really exciting. John Barros is getting it this year. Can we take a moment to... So I mentioned before about how, so the other thing I should mention is that she holds several records within our program. One is one of the first successful proposals to the NEI was, I believe, Woody, you were the PI on that one. Is that correct? Oh, it was Peter Ward? Who? John? Anyway, illustrious people here had one of the first. I think she was in the second group of winners and has the longest continuous funding in a large astrobiology project. So it's been, let's see, 15 years, 20 years, 18 years, for a total of something like $40 million. And again, pushing, I mean, she basically created the area in exoplanets that focused on astrobiology and the search for life. And astrophysics and astronomy would be nowhere without the work that she did in laying the groundwork for that, for those missions that are coming up. And so you may have heard of HWO, the Habitable World Observatory, which is the next great telescope that astrophysics is focused on. And I will just point out that she was involved in those early concepts. Most of her former students and postdocs are involved in some way. Jada is the acting or interim project scientist. Yeah, I have to get program and project right. And also someone she didn't mention that was also an alum from here was Sean Domigal Goldman, who was a postdoc with Vicki, and based on, in addition to her being an amazing scientist, she's also an incredibly well-organized individual. I mean, she gets stuff done. And he is now serving as the acting division director for astronomy and astrophysics at headquarters. So I have to thank her, and the community recognizes the contribution she's made in terms of training the next generation of astrobiologists ready to go. Actually, I'm hoping that they will be involved, and maybe their children will be. We don't know when these telescopes are going to happen, but yeah. So again, I want to highlight that she has been nominated for several teaching and mentorship awards, and we are so grateful. I mean, the whole community recognizes her contribution. We're so grateful for all the people that she impacted, including myself. I have to be honest that after the first seminar I saw her give, I thought, I want to go get another PhD. I mean, exoplanets, so cool. And she makes it so accessible. She also is a valued friend, and I'm really lucky to live on this coast and be able to see all of you and her, hopefully on a more regular basis. So with no further ado, please come up and after her talk, I'll go on to the second page of accolades.
Okay. I feel like I'm getting an award and not giving a talk, but thank you, Mary, so much for going through and giving such a thorough summary. Okay, let's move this in a bit. Make sure we are accessible here. How's that? Can you still hear me? No? It is. It's coming out of the speakers. Maybe you are, can you help with that at all? Oh, okay. All right. So we have a problem with assisted listening devices at the moment, but they're trying to troubleshoot it. Okay. All right. Cool. All right. Well, thank you, everybody. And thank you so much for coming to this lecture. This is the first of 4 lectures we're going to give to celebrate 25 years with of the astrobiology program here at the University of Washington. And these talks will be given every Wednesday of this month. So this is the first one coming up. And my talk is really setting the scene, introducing the topic, talking about astrobiology as a field, introducing you to the astrobiology program and what we've done, and also talking about the founding of our program, and then going into sort of the discoveries and the work that we have done with our students and our alumni. And so I hope I'll be able to talk about some exciting science highlights as well that we have done, and the way that we have pushed and changed the field of astrobiology, especially in the areas of the origin of life, and of course the search for life beyond the Earth, both in the solar system and out beyond our solar system into the exoplanet population. So astrobiology is focused around a central core question: are we alone? So the search for the answer to this question is really one of the most fundamental of humanity's goals and one of the most enduring. You can imagine our time as an early species sitting around the campfire, wondering if the stars in the sky were also campfires with other people sitting around them. We have now taken that concept over millennia and now are getting to the point where we have the telescopes that can help us look for these planets and potentially other life around them. So there's some very fundamental questions that astrobiology asks, and so I'm going to talk to you a little bit about these. I'm also going to sort of explain astrobiology as a field. I know when people first hear about the concept of astrobiology, they think it's a little bit science fiction-y. Some of the questions are, is this for real? Can you get paid to do this? You know, is this a career? And so I can talk a little bit about that and the fact that at the astrobiology program, we try and make this a career for every single one of our graduate students. And we have, in fact, been quite successful in that. So our fundamental questions are, so the first one we ask is, how did life begin and evolve on the Earth? And the field has advanced to the point where we're not just trying to smash things together in laboratory settings, though of course that's very important, but also trying to understand where did life first happen? What were the environments like? What were the characteristics of those environments that caused the ingredients for life, the building blocks to come together and to give us, sort of the first cells and the first replication? So that's a fundamental question in astrobiology. The other question is, do other planets in our own solar system harbor life? We know there's abundant life in our own world, and then we look out into the solar system and say, well, hey, there's no other things with surface oceans, but other environments where, maybe liquid water is seen underneath the surface of Mars, for example, or for the liquid water that we know is trapped in the cores of, say, icy worlds like Enceladus and Europa. Another question we're fundamentally interested in, I'm particularly interested in, is, do planets beyond our solar system, do planets orbiting around other stars, do they have the right conditions for life, the ones that we're finding out there? And so this is a question, again, about, are these things potentially habitable? Would life be able to survive on them? And later in this talk, I'll talk to you about how we even try and figure that out, given that these planets are so tiny and so far away. So we think about where these environments might be, and we ask ourselves, okay, how do we pick the best places or planets to go look for life? So that's really one of our fundamental things. And a lot of people criticize astrobiology and say, well, it's a science in search of a subject. You don't have any aliens. How can you possibly study life elsewhere? And the answer to that is we prepare. We try to understand, where would that life be? How do I find those environments elsewhere? If there's life in that environment, how do I tell? And most importantly, how do I disentangle the life's impact on that environment from the environment just being itself? And so that brings us to our final question, which is, how do we find and then recognize alien life when we go looking for it? So this fundamental question of, are we alone, is really one that is what we call massively interdisciplinary. And what that means is that we need lots of different disciplines, scientific disciplines primarily, to answer these questions about, what sort of science should we do to try and understand where we should look, to interpret what we're seeing, what kind of measurements should we take in the laboratory, in the field, on planets elsewhere. How do we do that particular job of trying to push humanity's understanding of our place in the universe out beyond the cradle of the Earth? And so the question then is, what is astrobiology? And so this is a definition of a field which is quite old, but I think it still stands. This is probably around when Mary and I were first getting into this. So astrobiology is the study of life in the universe, its origin, evolution, distribution, and future. So in its origin, we try to study the earliest environments on Earth and how they may have produced the conditions and processes necessary for the origin of life, and how that life could persist over time. We look at the evolution of how life co-evolves with the environment throughout Earth's history, because it's not just the life doing the evolving, the planet's evolving along at the same time, and the interplay between life and that planet as it evolves is very important to understand, because that really changes the outcome at the end. So we look for our environments as well at the limits of habitability, what we call extreme environments for life, because that helps us push out where we might be able to look for life elsewhere. We have a very nice ambient temperature and pressure and pH even in this room at the moment. We're surviving quite nicely here. But, if I put the temperature up or put the temperature down or change the pH or whatever, can life still survive in those environments? Because that might be very important, because those might be the conditions in the oceans of Europa or on the subsurface of Mars. So how far can we push life and its survival? And then one that I'm particularly interested in is this concept of the distribution of life. How do we identify and search for habitable environments elsewhere in the solar system and on extrasolar planets, and how do we best search for and identify life beyond the Earth? Okay. So astrobiology is not just the search for life elsewhere, it's all of these other things about understanding how life forms, how it evolves, what we should look for, where we should be looking. And then finally, we can think about the future. What is the future of life on Earth and beyond? What happens to us as a species? What happens to microbes as a species as we bring them out into other locations? So as Mary very nicely summarized, astrobiology is now incredibly important to NASA science and to NASA missions. We may have started out as something of a sort of a bit of an arcane field, but over the past 25 years or so, astrobiology has evolved to really underpin the fundamental science and many of the missions that NASA is developing. Some could argue pretty much all of them. And so there were documents, which Mary talked about, where the national academies put together community groups. We tried to understand, what do we need to do with exoplanet science? What should we do to search for life in the universe? Where is astronomy going in the next 10 years? Where is planetary science going in the next 10 years? And all of them have major themes: understanding worlds and suns in context, understanding the limits of life, and so on, that are really focused on this question of, is there life elsewhere? So if I go to the origins of astrobiology at NASA, NASA used to have an exobiology program, but the astrobiology program is more recent. Don't ask me to tell the difference exactly. You'll have to ask Mary about that one. But astrobiology came with its own institute, that I know. So astrobiology really started at NASA back in 95-96 with the twin discovery of the first planet found orbiting around a more solar type star. So that was the 51 Pegasus B planet. We astronomers had known deep in our hearts that ours was not the only planetary system in the universe, but we hadn't had proof. And so this was a shot across the bow that says, hey, planetary systems around stars like ours might be really quite common. And then the Mars rock, which was harvested from a meteorite field in Antarctica, blasted off Mars and landed in Antarctica, showed us that there were conditions within this rock that seemed to indicate that maybe it had been habitable at one time, that there had been liquid water and nutrients available. And then there was this intriguing scan that looked like it might, in fact, be a microbe. I think since then, the consensus is this is not necessarily a sign of life, but it certainly was a very exciting thing to learn about, at the time. So in 1998, NASA founded the NASA Astrobiology Program and the NASA Astrobiology Institute. I joined the Institute in about 2000, 2001. So I was around for a large fraction of the time as that body worked. It was sunset in 2018 because it had done its job. It had built an entire field around this field of astrobiology. So what about the origins of UW astrobiology? Well, we had all those things. We had 51 Peg and the Martian microbe, but also shortly after that in 1997, the discovery using magnetometers and magnetic fields of the body Europa moving in the magnetic field of Jupiter revealed that there seemed to be a conductor under the surface of Jupiter that was most likely to be a salty ocean. So that was incredibly exciting. So there's another ocean world in the solar system, not just the Earth, but Europa as an ocean world. And through that entire time in the 1990s, scientists were pushing the limits of life, finding microbes in extreme environments and trying to understand, how do you survive at 93 Celsius? How do you survive in purely acidic water? How do you survive in the cooling tanks of nuclear reactors? And so science was pushing sort of the limits of life throughout that time. So into this comes the UW Astrobiology Program. So it really started in the spring of 1996 when John Barris, who was here and is our Drake Award winner for this year, and Woody Sullivan, who's also here in astronomy, planned to have a Planets and Life seminar in the autumn. So in spring, they said, this would be great. Let's have a Planets and Life seminar. And in the intervening time between the planning and the seminar, the Mars rock was discovered, and people thought there might have been life on Mars in the past. And so Woody tells me that his seminar was packed. It was packed. So, I mean, that was really fortuitous timing that had happened. Realizing and having the vision to say, hey, this is a really cool area of study NASA might be interested in the future. In December 1997, a proposal led by Jim Staley, a microbiologist at the University of Washington, went to the NSF to get an integrative graduate education, research, and training program established at UW. And they were awarded that $2.5 million grant over 5 years to start this program in this incredibly exciting area. So in September of 1999, the first grad student cohort arrives, and we get started. And then in September 2001, we get 2 dedicated astrobiology professors, Professor Buick and Catling, arrive. And they're the new ones. I'm sort of an older one. I arrived in 2007, so I'm kind of way past that. And then in April 2001, Peter Ward led a proposal to NASA, I believe it was for CAN 2, to Mary's point, to provide funding from the NAI, again, for work here at the University of Washington. And then in 2005, Woody pulled off a real hat trick. He wrote another IGERT proposal. You don't usually get 2 of them, but we somehow did manage to get 2 of them. So kudos to Woody on that. And so that continued the program for another 5 years or so. And then I arrived in 2007, bringing about $7.8 million with me from my former institution, the Jet Propulsion Lab. And so that was also a way for us to fund the program. And then I was able to continue to get successful proposals one after the other to bring in funding that could potentially support this program via matching awards from the University of Washington. And then in 2012, I was able to lead the push, backed by the faculty, to introduce a dual title PhD in a home department and astrobiology to our program. And that was so popular that every single student who was in the existing certificate program for astrobiology transferred over at that time. So we had a mass stampede into the dual title PhD. And so now in 2025, we graduated 79, possibly more, interdisciplinary STEM PhDs, the majority of whom have gone on to faculty positions or leadership roles in NASA. So that's what we do. We train the next generation of faculty leaders and NASA leaders. But none of this could have been done without the vision of the founding members of the UW Astrobiology program. So Jim Staley in particular, who wrote that first IGERT proposal, helped by many colleagues, Peter Ward with his CAN, his NASA Astrobiology Institute proposal. Other leaders, of course, Woody there, who came in as the chair of the steering group, which is another way of saying the second director, although we didn't want to be called that. And Conway Leovy, who's sadly not with us anymore, and John Brownlee there as well in this photo. And I made the joke earlier that he's a bit like the pale blue dot. He looks like a mote suspended in a sunbeam in this particular shot. And then Jody and John, the power couple, so Jody Deming and John Barros, working in the area of extremophiles, Jody in ice, John at hydrothermal vents, understanding both microbes and viruses there. And in 2001, our new faculty, Roger Buick and David Catling, arrived, diversifying what we had in the way of support there. Now, there's a ton of us, okay? So this is the current faculty at the University of Washington Astrobiology program. I'm currently the 3rd program director with Jody, if she's still here, with Jody Young as my associate director. So we now even have an associate director, but we have, a very diverse, very dynamic group of faculty who now support this program. So that's not bad for thinking, that Mars rock sounds really cool. We should probably put a program out around this. Okay, so the UW Astrobiology program is really a pioneering program in doing this type of interdisciplinary training. We are the oldest and longest running astrobiology graduate program in the world. Thanks again to our founders leaping onto this opportunity within months of it actually occurring. We span 11 different departments and 4 colleges. If anybody wants to ask me how difficult that is to manage, feel free to come up afterwards. But we have to corral 11 different departments and 4 colleges into producing a uniform product for all of the students in our particular program. We offer a dual title PhD, we offer a graduate certificate, and we do a lot of interdisciplinary research because it's that research that trains our students. So I just want to put in a little personal vignette here. I mean, my first experience of the astrobiology program was coming on one of their field workshops, which are considered by our students to be one of the best things that we do. And so my introduction was I rocked up as a brand new professor in 2007, I think September of 2007. I was promptly bundled into an unmarked van and driven all the way across the state. And they took me to the channeled scablands, where we learned about all sorts of things, petrified forests, geology. I remember Roger standing on top of a rock and telling me about how glacier had dropped it just there. And so I learned, just a ton of immersive sort of geology throughout. But my best memory is of Woody forcing us all somewhat awkwardly to sing along with him, singing Roll On Columbia, Roll On, the Woody Guthrie song, on the banks over the top of the Columbia. And actually, it's a song about hydroelectric power, which I hadn't realized in the moment. So it's Roll On Columbia, roll on. Your power is turning our darkness to dawn. Roll on, Columbia, roll on. So anyway, very memorable, probably because of the awkwardness and the emotion, but certainly he did eventually get everybody to sing along, so that was great. Bonding on the trip. But that's also where I met Rika Anderson, who will be our next speaker in the sequence. She's coming next Tuesday. She is an absolute expert on getting in tiny little vessels and going down to the bottom of the ocean and understanding the deep ocean and what we can learn from it. So please join me in hearing about what she's doing too. So one of the main things that distinguishes our program from a lot of other things is the two things we offer: the workshops for students for interdisciplinary training, and then also their research rotations, which I'll talk about in a minute. So we've done astrobiology graduate workshops where we again take an astronomer and stick them on a boat here. You can see Jake Lestegege here on the Tommy Thompson. He looks the part, but he's an astronomer. Okay, and then we went to Death Valley. We went to Yellowstone. We went to Goodenough Lake, where we tried to lose a student. I'll talk about that later. And then this is one that I led single-handedly. You'll notice my broken arm. I led this thing single-handedly to the Jet Propulsion Lab. And I just want you to focus in on the students who are clustered around me here. This was their first time going to a NASA lab. Actually, that's probably not true for Lena, but for the rest of them, it certainly was. And I just want to say that out of that workshop, those people who were clustered around me, the 5 I circled, here they are. So Jonathan Babst is now working as the science systems engineer on the 2nd generation Mars Science helicopter. Elena Amador is managing Curiosity rover on Mars. She's also a science systems engineer. Anna Simpson is working in planetary protection. Osa Igbenosen, her wildest dream was to work at the Jet Propulsion Lab, and she, in fact, is a postdoc there. And then Jada, who Mary also mentioned, one of my students, went to the NASA Goddard Space Flight Center, and she is now the HWO interim project scientist, which is an enormous role for someone that early career. And I think the point of this was that the reason I took those people to JPL was I wanted to demystify and to reduce the stress levels on people considering working for NASA. People thinking, hey, I'm trained as an academic, I could be a postdoc. How could I possibly work on a NASA mission? And so we went there, we worked with the A team to help try and sort of brainstorm a mission design. They got to meet people who had just, stumbled into working for a mission coming directly from a degree or from a postdoc. And so it was a great experience for them to know that that was an option for them to follow. I also want to point out that, we also innovate not only in our graduate training, as far as the places we take them and what we do, but sometimes we have to innovate even within our workshop requirements, for example. And I just want to give you a quick overview of something that I thought was really remarkable, which was our astrobiology workshop COVID edition. When we were locked down, we could not hold classes. It was dangerous and difficult to shove people into a van and take them across Washington State. And so Dr. Brooke Nunn really rose to the challenge here. And she had been running, she's in genome sciences in our program, and she'd been running a CSI detective class where she stages a murder, leaves a bunch of clues around, and students have to collaborate all across campus and with others to try and gather the clues necessary and solve the murder. So I was talking to her about what we could do in the way of a workshop, and she said, well, maybe I could do something like that. I said, ooh, could you do something like that with like biosignature detection? She's like, leave it with me. So she came up with this amazing concept for our students to come together in a virtual workshop. They all worked out of their own homes. We sent them food. We figured we could at least do that. So they had to work to select instruments for a lander and orbiter to go to a mission. We had to design, and I think Rory, who's here, was instrumental in designing the planet to start with. So we had all of these sort of basic functionality of that. They had to collect and interpret raw data. So we asked them to pick a mission, pick your instruments. Once they picked the instruments, behind the scenes, Brooke and her absolute squad of grad student and postdoc elves ran around generating fake data for these missions. And then the students analyzed the data and tried to assemble evidence as to whether or not this planet had life on it or not. So in the process, they learned about instrumental capabilities and limitations, mission-focused experimental design, what could trip you up, not having enough samples, not doing controls or replication, how the environment of the planet really shapes your ability to interpret what you're seeing, and how you should critically think about, how do I separate planetary processes from life processes? How do I convince people that I really have life here? And so they got a chance to network with their peers. And because it was COVID and because we were all on Zoom, we were able to get a whole series of amazing experts from around the planet to come and talk to our students about the missions that they had developed and what they had done. So that was really cool. And so we had the mission timeline. So Brooke cracked the whip, and these students had to design a mission in 4 days and then come up with the measurements and the arguments for whether or not they thought life was on it. And on the final day, they held a debate. And this was the debate going on here. Mary, I think Mary, you actually attended the debate. So we had outside people also attend the debate. What we discovered was it was really hard to convince anybody that you had detected life. So that was a valuable lesson to learn. But headquarters really loved it. And I won't go through all the text here, but they said, the attention to detail, the way that the process was done and what people learned in the process was just really amazing. And the students and postdocs also loved it as well, giving them a chance to network across disciplines and consult with each other and with other experts. And so, NASA held this up as an example of like an amazing thing that we should be doing, and then put their money where their mouth was. And in fact, asked Brooke to do another one, but in earnest, to try and design a mission to Mars. For example, to understand as we're designing the mission, what do we have to think about if we want to try and detect life with this mission? So that's one of our workshops. Research rotations, again, our students get to go to amazing places. The whole point of this is to go and work in a lab outside of your own, learn a new technique, do something different. It could be completely different, or it could be somewhat parallel to what you're doing, but go and learn and network. And these, again, are activities that are incredibly important. So we had popular science books as well, textbooks and science reviews that we have written as a group that I just wanted to call out. There's Peter Ward and Don Brownlee's Rare Earth book, which was incredibly popular. I hear from Peter that you guys are writing Mark II or something at the moment, so people can look out for that. There was the Planets and Life book that came out of that original seminar, which Woody and John Bowers put together. John's also in charge of a National Academy's report that is called The Limits of Organic Life in Planetary Systems, but is known colloquially as the Weird Life Report. And it is simply about how far can we push life before it breaks. Dave Catling has written a nice and short introduction to astrobiology, if anybody's interested in that. And I and Jada edited a planetary astrobiology book to try and bring astrobiology to planetary scientists. That is a more technical book. It is for scientists, but again, we tried to write it so that it was accessible to others. We've done some really cool and unusual outreach, and this was something that Woody worked on with Dr. Glenna Burma, who donated the funding to allow this to happen. But we got together as astrobiologists with a bunch of composers and talked about our science and talked about what was important. And they composed a symphony based on what they had learned, complete with graphics and everything else. That symphony occurred in 2015 in Benaroya Hall. But if you are at all interested, we do have a link there to the Vimeo recording of that particular concert if you'd like to take that and have a look at it. Or you can have a look on our website and we'll get this up as a news item. Anyway, our astrobiology graduates do have some absolutely amazing careers. Some of them are listed up there. Many of them have to do with NASA. A lot of them are also faculty positions as well. I wanted to point out we do have our speakers coming. So Rika next week, Ken, who's in the audience, will be coming the week after that to talk about Mars 2020. And then we have Omar Shields coming to talk about her experience and her science as a student who's come from this particular program. We were hoping to have Jada here, but because she is a federal employee, she is not permitted to travel at this time. So unfortunately, Jada will not be pairing with Rika to deliver the talk we had wanted from her. Our students also go out, some of them are entrepreneurs, they work for the Washington State Department of Natural Resources, they have a whole bunch of incredibly interesting careers. And I will point out that one of our students now, in the first cohort, is a full professor. So that's how old our program is. We now have a full professor alumnus. And I also want to point out this guy here, Randall Perry, he's the CEO of Space Science and Engineering Foundation, but he also writes novels. I learned a lot researching this talk. And so he writes thrillers where there's an astrobiologist who is the hero, who solves mysteries. It got, I'm not getting any royalties, but it got 4.5 out of 5 stars on Amazon, so it's not terrible. So I think, you know, if you want to hear a story where an astrobiologist is the hero, and I certainly do, so I'm going to go buy a copy. But yeah, so we've done a bunch of things there. Okay, so let's talk a bit more about the science then. So at UW Astrobiology, we have five major research themes that we're interested in: the origin and evolution of life on Earth, life in extreme environments, habitability and life in the solar system. These sound familiar. Exoplanet detection, habitability and biosignatures. We do a lot of that. And space exploration. So I'm going to talk a little bit now about the science that we actually get to do and highlight the research that's being done by our students, our alumni, and our faculty. So the question of the origin and evolution of life on Earth is just a huge one. I mean, the fundamental question is how and where does life originate? And the how has been very much a focus of the origin of life, but the where has not been paid as much attention to. But that's certainly changing within the field. And so we're now very much interested in, well, I managed to artificially whack these things together in the lab, but, you know, would that condition happen elsewhere on the planet? Can I plausibly say that this is an environment that life might actually be found in? And so I got this great quote while I was looking up the origin of life from George Whitesides on receiving his Priestley Medal, which is the highest award in chemistry. And he said, "The origin of life. This problem is one of the big ones in science. It begins to place life and us in the universe. Most chemists believe, as do I, that life emerged spontaneously from mixtures of molecules in the prebiotic Earth. How? I have no idea." Okay. So this is certainly an incredibly challenging field. It can get quite heated, simply because there isn't that much evidence. You'll find that in scientific fields, the less evidence, the more passionate people are about their positions. Okay, so you've probably heard about Darwin's warm little pond, that's where he hypothesized that maybe life would have occurred somewhere in some warm little pond that, you know, created, that had phosphates in it and potentially ammonia, that would be an environment where that could happen. There is a problem with that, though, in that his warm little pond probably wouldn't have produced the building blocks of life as we know it. I mean, part of that is because his warm little pond would have. Whether or not that warm little pond existed is another question we have altogether. So he imagined a place where there was ammonia, where there were these compounds that created, had a lot of hydrogen within them. And certainly the first Miller-Urey experiments, if people have heard of those, that's when ammonia and methane was put in a chamber and zapped with lightning, and it produced organic compounds. But we now know that the early Earth atmosphere was probably outgassed from volcanoes and more CO2 dominated. It would have been hard to get these types of reactions under those conditions. We also have to ask ourselves, okay, but how were amino acids linked into chains of polymers in this particular situation? Especially if we want to talk about life at hydrothermal vents, for example. Water and heat near hydrothermal vents can make it difficult to link these things together. How do we overcome that dilution? There's just lots and lots of challenges. For each thing we come up with, is kind of there's a problem with that. And then to try and have this warm little pond with the phosphates in it, to make prebiotic molecules, you do have to have like an incredibly high concentration of phosphate. And until recently, there wasn't really anywhere on the planet, at least, that had that particular characteristic. So these are some of the problems with the origin of life that our people are tackling. So, and this is again really interesting because this was a class that was run by John Barris, one of our winter seminars, and in it a bunch of our fabulous alumni, who are now alumni, who were students at the time, asked this question: where is the optimal environment for the origin of life? And they came up with the conclusion that actually it wasn't a particular place on the planet, it was the entire planet. That we had to think of the origin of life and environment as being essentially a global chemical reactor. That things could be made in this environment, or this other thing could be made in that environment, and we'd have to transport them all to get everything together. So this was published after this class, and I think it's got like 150 citations now. It's actually pretty popular. So that was sort of an intriguing way of getting at, do we have the right environment? When trying to talk about, okay, well, if I've got outgassing and I can't make my primordial hydrocarbons or whatever, how do I get my primordial soup? And so Nick Wergard, working with Dave Catling, came up with the idea that maybe I just smash rocks into the earth, and in the aftermath of those impacts, I can generate a lot of hydrogen-bearing compounds that can ultimately be used to create that primordial soup that we need. And so it's possible that the evolution of that organic primordial soup was due to punctuated evolution, in a sense, from impacts happening. So that was an intriguing paper. Then we have another one. This is from the chemistry lab of Sarah Keller, working with Roy Black as well. Roy, I didn't put your photo up there, I apologize, but Roy's in the audience also. And this was kind of a breakthrough in trying to understand, well, it was a breakthrough, in trying to understand how we get not just sort of protocells forming, but forming in the presence of things that we could use to make life, like amino acids, and overcoming the issue that when you try and form protocells in a salty environment like the ocean, or somewhere where you might think life might try and start out, that salt and also magnesium cations, for example, will disrupt the formation of the cell. So how do we get past trying to form our cell, having it disrupt or flocculate into little tiny pieces, et cetera, et cetera? And what they discovered was that if you actually put amino acids in with your salty compounds and the fatty acids you need to make this protocell walls, that they all kind of glom together and they stabilize each other. So that was important because it showed that we could stabilize these early protocells as long as we had amino acids present, RNA. And also that this is one way they could have all started hanging out together and potentially reacting, the early Earth. So this was a pretty exciting discovery. It became the most read article in the edition of The Atlantic, in which Ed Young wrote a story about it. And also, I don't know this person, but I was told younger people do. But Hank Green from the Science Show apparently also really featured this as well. So that was a big breakthrough. Sorry, I'm quite old. So the other problem I talked about at the end, this idea that we had a warm little pond with the phosphorus, the phosphate in it, and that there wasn't really a place potentially where that might exist. This Dave Catling sent me this slide, and he said, tucked away in tables at the back of a 1995 master's thesis from the University of Saskatchewan, I found that the phosphate levels in Last Chance Lake are so high that they might actually be, a good spot to go look for the origin of life. Environment, right? Not life happening in front of us. So Dave and John and more of his team were basically like, okay, well, this could be a fantastic location. Maybe we can find something that is a modern day analog of an environment in which life evolved on our planet. And so not content with just going on a science trip, David also bravely volunteered to take a whole bunch of our students there as part of a workshop, one of the workshops I kept talking about. So unfortunately, we nearly lost Zach Cohen there, which would be unfortunate because he wrote this really great paper afterwards. So here's Zach sinking. Sebastian Haas is carefully watching him sinking, wondering what to do. But luckily, the professor steps in. So David steps in and hands him a shoe. Now, I don't know where the shoe came from because both David and Zach are wearing waders, but somehow the shoe magically appeared. And maybe you can explain to us where that shoe came from. But hopefully that shoe actually saved Zach because he was then able to write this paper afterwards. So there's Zach, unharmed. But yeah, apparently the top of these lakes crusts over like creme brulee, the sugar on top of that dessert, and you can put your feet right through it. But Zach was able to show that actually the soda lakes did have the types of environmental constraints, environmental characteristics that would allow the sorts of experiments that Sarah and Roy were doing to work. And so that this was, in fact, these soda lakes, which are high in sodium and carbonate, might actually be a good analog for where life might have occurred. And then Sebastian went on, the postdoc who was watching Zach sink, Sebastian went on to study sort of the biogeochemistry of those areas and try to understand, okay, how do you get phosphate to build up that high? And he concluded that, you know, because of nitrogen deficiency, which meant that there was a lack of life there, which is good if we're trying to find an analog site for the origin of life, right? That these lakes had a lack of life in them, and also just the way that the calcium was precipitated meant that phosphate was able to build up continuously in there. So we learned a bunch of things. Potentially a good environment. We can run the experiments we ran in the lab with this type of water, and then also we kind of understand why that's happening. Okay, so this one, let's step out from the origin of life. This is really one of my favorite things that's come out of the program. So this was work that was led by Sanjoy Som, and Sanjoy was an engineering student for like 6 years before he came, and I think it was one of your classes, Dave. He came and he listened to Dave talking about planetary science and astrobiology, and he said, you know what, I want to do that. And so he changed his degree over to ESS, but he brought his engineering background with him, which is something we love in our program. And so he figured out ways to actually try and I think Roger Buick probably had a big hand in this, figured out ways to try and measure the density, the pressure of the atmosphere like 2.7 billion years ago. And the way he did that, there was 2 ways to do that. One was, you can see this adorable meerkat licking these little sort of indentations in this rock in South Africa. And these are in fact fossilized raindrop imprints from rain that has fallen on volcanic ash in a riverbed that was then covered over. And the thing is, with the pressure of an atmosphere, that changes sort of the terminal size of a raindrop that you can have, and therefore the maximum raindrop size you see impacting. And so by measuring the size of the maximum size of the raindrop distribution, he could figure out some constraints on the atmosphere. He showed that it was probably less than 2 times what we have right now, which was a bit of a shock because people were desperately hoping that the atmosphere was much thicker in the past to try and explain why we hadn't all frozen over because our sun was fainter back then. So that didn't help, but truth is truth. So, and then the next thing he tried was looking at something called pillow basalts, which are volcanic sort of, you know, as the lava flows down to the sea, essentially lets us know, I believe it's lava toes or something. There's a piece of the actual lava emplacement that lets you know that it hit the ocean at some point, and so therefore this thing is at sea level. And by looking at the size of the vesicles, the size of the bubbles in that lava over time, you can figure out the pressure of the overlying atmosphere in there as well. And so for 2.7 billion years ago, he was able to refine his measurement to about a quarter, plus or minus a quarter, the pressure we currently have on the surface of the Earth. And so definitely not a thick atmosphere back in the Neo-Archean. But isn't this cool that you can take fossilized rocks, showing geological processes from the past and using some physics, you know, figure out what, you know, constraints on that early atmosphere. Okay, so life in extreme environments. This is another big area that we do here at the University of Washington. So the questions here are, you know, what are the environmental limits of life on Earth? How does life survive in these environments? I mean, how does it manage to do this? And what does that mean for how and where we should search for life elsewhere? You know, if we find something that can survive in conditions that look very Mars-like, well, maybe that's something we would look for on Mars. So here we have what I think is one of the most kick-ass photographs to ever come out of our program. And so this is Jeff Bowman on the ice, and this ice is thin. Jody tells me it's what, a few inches? A few inches over thousands of meters of icy cold Arctic Ocean. And so Jeff is kneeling on the ice here, and he is collecting something called frost flowers. And so frost flowers occur when water from below the ice is actually wicked up and creates these structures on the ice. And the interesting thing about them is they tend to concentrate nutrients. You tend to find a concentration of bacteria growing in these particular frost flowers. So they're kind of an extreme environment, but they're colder, I believe, than the surrounding area as well. So, you know, how habitable is the ice on Earth is the overarching question that's asked by Jody and others in the oceanography department. And how and where should we look for life on icy worlds? So these frost flowers were studied and showed to contain a very high salt concentration, which normally isn't good for life. And also a lot of this extra polysaccharide, EPS, which is something that Jody and her group has shown is given off by microbes when they're in ice, and it's like an antifreeze. It creates a little bivouac of unfrozen water around the organism to allow it to survive in freezing conditions. So there's a ton of that in there, lots of salt, high bacterial concentrations, and also some interesting things that I want to follow up with Jody after this talk about how these things interact with the atmosphere, what they give off, what they draw down, because that could get me a biosignature too. Anyway, other work that Jody and her team are doing, this is Zach Cooper. I don't know what Zach did, but he was sent down this ice hole in the Arctic. I guess he just had a lot of questions about the universe. So he sent down his ice hole in the Arctic, and this is drilling into something called cryopegs, which is salty water that is preserved, isolated really for tens of thousands of years at the base of these structures. And the astonishing thing about these is that they are very heavily inhabited. And so there's lots and lots, not a lot of diversity, but there's lots of organisms down there. In fact, maybe I believe more than in the surrounding ice or other environments. So that's pretty fascinating because that's kind of an analog for what we might find on Mars. So here we are pushing the limits of life and trying to find out what's going on. At the other end of the temperature scale, hydrothermal vents are also a big thing that people look for. And this is this work that was led by, I would say, Deb Kelly and John Barros, and then Billy and Brooke are also working in this area. But Deb discovered in 2001 this Lost City hydrothermal vent field that isn't heated by magma coming up from below. It's in fact heated by a reaction between water and rock. And that water and rock reaction is endothermic. It gives off heat. Exothermic. Got that wrong. Gives off heat and also produces methane and hydrogen at the same time. And so it's an analog potentially for subsurface water running over ultramafic rock on Mars. Also potentially this process could be happening at the bottom of the European Ocean, and it might have been quite common on the early Earth as well. So these are great sites to try and understand what's going on, what's there, what lives there, what is it producing, and so on. And in 2009, John and colleagues proposed that hydrothermal vents might actually be a possible location for the origin of life, and that's part of his Drake Award citation as well. Okay, so pretty cool. No magnet involved, just water and rock. All right, and so in another great example of cross-departmental collaboration with our group, Professor Corbin Lewis in ESS is actually studying an organism that John isolated from a hydrothermal vent as part of his hydrothermal vent studies. This organism can metabolize nitrogen at 92 Celsius. Don't know about you, but I'm not really good close to the boiling point of water. And so this is the highest recorded temperature for this process. So the team is trying to understand sort of the energy that this organism can produce when it's doing that nitrogen metabolism and when it isn't, to try and sort of constrain the habitability of environments for organisms that can and can't do this type of process. Okay, habitability and life in the solar system. I don't have a huge amount on this because Ken is going to be talking, I am sure, about a ton of stuff to do with Mars, but I will just say that basically Dave Catling was a member of the Phoenix Lander team and was able, with that data, working on that mission, to actually prove the hypothesis that the Mars Odyssey had made using remote sensing maps. It said, you know what? There seems to be a lot of ice under the very surface of Mars. And so Dave sent me this photo where they actually got a photo of the ice under the surface of Mars and were able to show that that's what was happening, but they also found an abundance of perchlorate, which is something that can be formed in the atmosphere, as he showed. And so we're seeing sort of some of the processes on Mars that are actually affecting the near surface environment and therefore, an area where life could potentially be. Okay, I realize that my timing marker has been stuck for a while. So I will now talk a little bit about the research that my own team does. Then I will try to wrap this up in 10 minutes, I think, given what we have here. So the VPL, the Virtual Planetary Laboratory, is an interdisciplinary consortium of about 60 scientists at 20 institutions. That's also a lot of cats to wrangle or to herd in a broader space. You can see here that we actually became part of a NASA cartoon book, which is probably one of my greatest achievements, is that we finally made it into a NASA cartoon book. These are available, by the way, on the NASA site for now. So feel free to, no, seriously, I got some 404 links when I was looking for things. So yeah, please feel free to download these. These are free. They're great. So yes, we can learn a lot about astrobiology, but in graphic novel form. So we pioneered, and Mary talked about this, we pioneered a bunch of key themes in exoplanet astrobiology. We have Earth as an exoplanet. We really study that to death, and we'll talk a little bit about that. Early Earth, as we realized that early Earth was a series of alien but inhabited planets, and then we could try and understand that. As Giada so famously and poetically said, early Earth is an alien Earth for which we have biological and geological constraints. And so we've studied those over time. We have looked at star-planet interactions and what that does to, whether or not we can see a biosignature on an extrasolar planet. And we've really pushed this point. It's probably one of the things we give to the field of astrobiology is this idea that interpreting potential signs of life must be done in the context of the environment in which you find that life. That is not enough to just say, I'm going to go and measure oxygen. And if I find oxygen, then therefore there's a biosphere on this planet. But realizing that oxygen might be formed by other processes as well. Okay, so how do we search for life on exoplanets? There's a bunch of things we need to do. First, we have to find exoplanets that are likely to have the right conditions for life. And then we have to figure out how we would recognize life on those planets if we were able to do that. So the community, the exoplanet astrobiology community, sees two opportunities for us to try and find life on extrasolar planets. One in the near term in the 2020s and one in the longer term in the 2030s. The near-term one is to look at planets that pass in front of their star. So this is transmission or transiting planets where we see the atmosphere of the star, the atmosphere of the planet backlit. Those planets will preferentially be around M dwarfs. You can ask me why in the question session, but that's the way it works. And so we can potentially look for life then with JWST and ground-based extremely large telescopes. And then the other way we could do this is in the 2030s, we're hoping for the launch of this spacecraft, the Habitable Worlds Observatory, and it will be able to directly image planets orbiting around other stars. And by doing that direct imaging, we will be able to get a spectrum of that planet and learn more about it. So two ways, either passing in front of the star like this, or like Venus, as you can see in this particular shot here. You can see Venus off the edge there. This is a shot of Venus transiting the sun, but I've also got my little extrasolar planet passing in front of its star in the front here. But I will say that these transiting planets, this is one of the innovations that we have really pushed here in this field, and this is work that was done by Professor Eric Agel in the astronomy department, is that you can actually weigh a planet to amazing and exquisite precision by watching it pass in front of its star and timing how long it takes to come back to pass again. Because if the orbit of the planet was perfectly circular, it would go around, say, in 2 days exactly, and it would come back in 2 days, and it would always be 2 days. But we see planets that come back in, a tiny bit less than 2 days and a tiny bit more than 2 days. What's going on? What's happening is that planet is being tugged by other planets in the system. And so that's what we call transit timing variations. That tugging is gravitational. By using that gravitational effect, we can actually pull out the masses of the planet. And by doing that, we can therefore get the mass of the planet, its size from the transit. We can start looking at the density of the planets. And one of the most spectacular plots I've ever seen is this one, which those little colored clouds that are going along that arc there are showing the density estimates for planets in the TRAPPIST-1 system, a system of 7 Earth-sized planets orbiting around another star. And they pretty much all land on the same line. It's telling us that, unless there's sort of some interesting things going on, that these things basically have a similar density to each other. They were potentially formed in the same way. They're potentially made of the same stuff. They seem to be very similar to what we find for the Earth. We've also done work looking at trying to understand. This is another thing that VPL does, is like we go into a problem that's really, really hard and we just like hammer it. So here's another one, which is like, okay, I have a planet that has liquid water on its surface. How do I get that liquid water? How do I maintain it? I have to look at the stellar effects on the planet. I have to look at what happens to the planet and its planetary system. Is it being tugged around in its orbit? Does it have the gravitational influences? And I have to look at the planetary properties itself. And Rory Barnes and I, two professors here at the University of Washington, figured out that, of course, understanding what is actually going on with all of these processes is really important for understanding whether or not a planet is habitable. And so we held a workshop in 2010 where, as a community, we sat down and tried to figure out, okay, what are all the things that affect whether or not a planet is habitable? And we came up with this diagram, which is colloquially known as planets are hard. Okay? Planets are hard. So, yeah, but I mean, it really caused the field to sit back and think, oh, how do I measure all of these particular things? That's quite complicated. Other things we've done is looked at the evolution of planets around their star, and in particular shown that the stellar evolution of the star can in fact change the evolution of the planet, and in such a way, and this was also another very influential paper done by one of our UWAB graduate students, Rodrigo Luger, and his advisor, Professor Rory Barnes, which just showed that some planets around M-dwarf stars in particular will spend a large fraction of their early life being way too hot because their star is way too bright. It dims as it comes down into the main sequence. And so that means that we can drive ocean and atmospheric loss very early on. Does the planet recover? We don't know. So, I mean, this is a really major, major question about life around other, life on planets around other stars. This is work that was done by then graduate student Andrew Lankowski, who is also here, but just looking at, okay, what does that mean for those TRAPPIST-1 planets? Those 7 planets I told you about that are Earth-sized? What happens if they turn into Venuses? If they're way too hot to begin with, are they going to be stable over time? What happens to their surface temperature? And what we're really showing with this diagram, don't worry about the details, is that all these colors indicate a temperature. And over here, these things indicate a different composition for the atmosphere. Is it mostly oxygen? Is it mostly CO2? What's it made of? But the point is, each of these planets are all sitting in the same position relative to the star. Depending on their atmospheric composition, they could be amazingly different temperatures. And so it's not enough to just say, oh, this planet's this far from the star, therefore it must be this temperature. We really need to understand what it's made of to be able to understand what's going on here. And then I think, and I will try and skip through some of this here, but I think one of the main things that we have done is this concept of understanding that there are false positives, that there are ways of generating a biosignature gas using a planetary process that has nothing to do with life, and that it's much more prevalent than we thought. So when I came into this field initially, everybody said, oh, all you have to do is measure oxygen. That means we have an abundant photosynthetic biosphere. Oxygen is it. It's great. So if you get methane as well, fantastic. But, and what we discovered, and this is what I call science biannual report. So you will hear a lot of people like me at PI say, oh my God, the annual report, I have to write the annual report. But what happened was, while we were putting this annual report together, I discovered that like 4 or 5 separate subgroups of my team had actually come up with different ways of making oxygen without life. And so I realized that, oh, we're sitting on a really big thing here, because everybody assumed that oxygen would just be an easy thing to go and look for on a planet to say photosynthesis is happening. And so what this diagram is actually showing is a bunch of different conditions under which oxygen could be generated without life, but it's also showing the tells. It's showing the things that are happening in the environment that let you know that the planet made the oxygen and not life. So that's what's called a false positive indicator. Okay, so I'm going to skip through some stuff here to finish it in a reasonable amount of time. And so one other thing I just want to emphasize is this idea of the Earth as an exoplanet and trying to search for alien oceans. So I've gone from looking at things in transmission with JWST to thinking about, okay, if I have a habitable worlds observatory image of a planet around another star, how do I tell whether or not it has an ocean on it? How can I actually do that? And so this is basically Mary alluded to this. One of the things that we do, because I was trained as a planetary scientist in our solar system, so I'm always looking around for, do we have an example of that somewhere that we could use as an analog for an exoplanet system? And so you can see circled in red here, this is an image of the Earth taken from the L-Cross spacecraft before it crashed into the moon. And you can see that we're in crescent phase. Can you see that dot of light on the edge of the crescent there? Like in the middle, looks like it's a ring almost with I guess, a diamond or something in it. That is glint off the ocean of the Earth. And in the crescent phase, it's incredibly bright because of the angle that the radiation is, the sun's light is coming in and bouncing off the ocean. And so we were able to see that with the L-Cross data. We could see this effect in the crescent phase that is not as obvious when you have closer to a full phase of the Earth. And so we were able to actually use these to try and understand how detectable it would be to try and see that glint in the crescent phase and to be able to look for oceans that way. And I will say another example of doing this sort of solar system exoplanet comparison work by using these sort of analog things is Ty also discovered that there was an error in the calibration for the data being used for the spacecraft to understand the moon. Because they sent us a spectrum and they said, well, this is our spectrum of the Earth. And Ty said, yeah, no, it's not. They're like, no, it is. And we're like, well, that's really odd because it's not supposed to look like that. So in the end, we figured out, oh, there was actually a sinusoidal error that hadn't been taken out in the data. So that's not a terrible thing. It's fantastic. We were able to help them improve their calibration and therefore improve the data from the mission itself. So these sorts of comparisons are really, really important. And so, yeah, just one other thing, since Jake is here, I'll go over this. So Jake Lustig-Jaeger then took sort of this idea of looking for unnatural brightness in the crescent phase due to the glint, and he did a better job of this by mapping a planet as it rotates underneath you. You can actually separate out, oh, here's one surface and here's another surface. And if you look at the change in brightness of the surface when it's near crescent and when it's not near crescent, this orange thing here, the two lines are on top of each other. The brightness doesn't really change. But this blue thing here, the brightness changes hugely between when it's in crescent phase and when it is not in crescent phase. And so he was able to separate out the ocean itself and the effect of it, which just gave a much better sensitivity to this phenomenon actually happening. Okay, and so I think, given the time here and the fact that my cursor wasn't running, I'm going to move finally to space exploration. I just want to say that we, as a faculty and with students and alumni, just do a ton of stuff for spaceflight missions. It's one of the things we really enjoy and the things we really get into. And I want to also, we do some really interesting things. So that the chair of psychology came up to me and said, what is psychology doing with astrobiology? And I said, well, there's a slide. So this is work done by Dominic Civitelli, who was a student in our program. He studies octopus cognition in the Department of Psychology. In particular, how they sense their environment, how they get things and retrieve them, and so on. And he, because he went on a research rotation to a robotics firm, is now working on basically a robotic octopus that could potentially be used in exploratory areas, like going down, for example, I think the grand dream is to go down, one of the chasms on Enceladus, and like an octopus, find your way through small spaces and so on, to be able to do that. So that's kind of a pretty cool example. So what's next for UW Astrobiology? We'll just wrap this up. Well, we did this Mars workshop that was based on our COVID workshop with the students. And a bunch of us here helped out with that. These are all the people who helped with that. And we've been asked to propose to do another one for Icy Worlds. The fun thing about this is it helps NASA in mission design and with instrument payloads by improving development of the final mission by running through and making mistakes before we build anything. Running through the simulation to see if we can break it, to see what information we have missing, to see where the ambiguities are before we start cutting metal or even designing the spacecraft. There's another mission that's just recently been funded, but I don't think the funding's here yet, but it's coming. It's called BIOS Endurance. This is a collaboration between, I should put everybody's names down, but I didn't, but this is a collaboration between aeronautics and astronautics, oceanography, School of Environmental and Forest Sciences, ESS, and Genome Sciences to look at the impact of stressors in space on both humans and plants. And so we're very much interested in looking at that. And then finally, the Virtual Planetary Laboratory was funded for another 5 years. And we are working on understanding how to predict and to interpret the information that will come from the Habitable Worlds Observatory, and to get a statistical framework under telling whether or not we think we're confident we have life. So I'm finishing there, even though it says, but wait, there's more. If you're interested in any of this astrobiology stuff and you're not in our program, please feel free to join our colloquium series. We have it every Tuesday at 3 p.m. in spring and autumn. We also record it. It's archived on our website. So if you want to hear sort of more detail on any of these sorts of things, feel free to browse through our YouTube channel and check that out. You have now successfully completed April 2nd on your punch card. There's another 3 of these to go. And then finally, of course, we always end with this one. So we could still use your support, especially in funding students to go on research rotations and workshops, which is a very expensive part of our program, but it's just extremely, extremely valuable. So I will leave it there, and we can have some questions.
All right, thanks everyone so much. And yeah, if you have to go at this point, yeah, you're welcome to sneak out, but we will do some questions at this time. So I'm trying to unravel the microphone right now. Do you want to stand up here at the podium? Or do you want to sit down?
I can both stand a little longer.
We have another one. If you want.
Okay.
Okay.
Questions. Oh, there's questions at the back there. And here.
Yeah. So I'm Virgil. Half the reason why I came to this school is I want to go into astrobiology, and this is a school that has an awesome program, and I love UW, so it's like hell yeah. So I guess my question is
You mean microphone?
How do I get into your program, and yeah, 2, how do I improve my chances of getting into it? Because it seems like there aren't that many students in it.
Okay. Yes, certainly. So what's your area of expertise?
Biology.
Biology. Oh, cool. That's great.
Yeah,
okay.
biology, and I also
yes, so getting into the astrobiology program, yes, so you would have to apply to graduate school. And the thing about the Astrobio program is we are not standalone because of the way our degree is set up. You have to do a home department and astrobiology. Mm-hmm. So you have to get into the home department first and apply to the astrobiology program. And then if you get into your home department, we can put you in the astrobiology program. So that's the first challenge. The first challenge is getting into the home department.
Yeah, do you have to wait until the home department accepts you in order to apply for the Astrobio, or can you?
No, and you can go ahead and apply in advance. That's fine. But we will typically not offer you a letter of admission though until the home department says they're taking you on, because we can't. The way it's set up, I won't bore you with the mechanism, but yeah, you have to be enrolled or intending to be enrolled in one of the regular departments to be in our program. But you can absolutely go ahead and apply at any time.
Gotcha.
Oh, yes. There's also an Introduction to Astrobiology, AB 115, which is taught by our various professors. So if you want to learn a little bit more about astrobiology, it is an introductory class, but it provides a really nice overview of the field.
Awesome.
Yeah.
I guess the other question was, so how competitive is it in terms of being able to get into the program?
It's generally no more competitive than it is to get into grad school at the University of Washington. So, so it's pretty competitive. Sure. So yeah, you do have to get into your home department. That is the major hurdle, and then after that, it's usually pretty straightforward. We don't typically, at the moment, get more astrobiology folks than we can take, based on the bottleneck that is the home department.
Cool.
Someone who's gone through this process, I would also just say, go knock people's doors down and ask them to join their research. And you can do that as an undergraduate.
I imagine that networking with the Astrobio department, before and during application would probably help, right?
Could do, yes. I mean, again, we're relying on the home department's the home department's admissions program though. So that's the first thing. But certainly come to our colloquia, come talk to us. I mean, the more you can learn about what astrobiology is and what interests us, the better your application will be.
Do research. Yeah, other questions? Yeah.
There's one at the back too.
So I get this question all the time, so I'm going to ask you.
And you can speak on behalf of UW's Astrobiology group. So all the wonderful potentially habitable environments that we have within our solar system and beyond, where do you think we're going to find life?
Okay, so I mean, this is something we always set up as a debate at AbSciCon, right? It's like so the question at AbSciCon is usually, will we find life in the solar system first or in exoplanets first? I can't answer that question, but I can explain why it's hard to answer. And it's hard to answer because in the solar system, we have all these fantastic robotic spacecraft, and we can go to places and we can drill them, and we can, get fluids, and we can measure with 5 or 6 different instruments. And so you have a chance in a limited number of environments that are probably quite harsh, and so you would only expect extremophiles. You can make detailed measurements and get more confidence on whether or not you're seeing life. For beyond the solar system, I like to make the joke, you can have any data you want as long as it's a spectrum, right? So we can only take spectra of these distant worlds. We're doing remote sensing. We can get photometry as well, but it's usually never as good as spectra. But we can get these spectra of these distant worlds, but then we're in the process of trying to disentangle life from an entire planet, a spectrum, an entire planet. So that's much more challenging. So either way, but on the other hand, the advantage for us is we're detecting, we're studying planets that potentially have liquid ocean water on their surface. And so therefore the biosphere is right there interacting with the atmosphere, which makes the signals much bigger. So in the solar system, you have to dig down. Maybe there's a small number of bacteria or whatever that you're trying to get at. Lots of different ways of getting at things, but low probability there's something there. Ocean world, surface ocean, entire planet like the Earth, maybe a higher probability of life, but just so hard to interpret. So which one will we find first? I don't know, but I'm looking forward to finding out.
Hi, my name is Kirti. I'm a working professional as a senior machine learning engineer at Fortive, and I am really interested in astronomy since childhood as a domain. It fascinates me a lot. Now, I want to know if UW Astrobiology department has seen people like me with an engineering background, probably science background, data science background. With that background, how is the acceptance? And yeah, I mean, and probably like prerequisites if you want to get into it. Yeah.
Right. So you would need to come in at the graduate level. I see Christy's just left, but we just had the chair of the aeronautics and astronautics department here, which is sort of one that is affiliated with the astrobiology program. So if you're interested in aeronautics and astronautics and you can get into their grad school, then that's a direct path into the astrobiology program to do it that way. I mean, as I said before, Sanjoy came in to us with an engineering degree, or most of an engineering degree pretty much already done. So we love engineers. I mean, Ty Robinson, the person who's doing the Glint stuff, he came from an engineering background as well. So again, we need everybody across the board, different backgrounds, different training and whatever to help in this really huge question. So, I mean, I don't think we would discriminate against someone with an engineering background because you know how to solve problems, you know how to work within parameters. I mean, it's a really great training to have. And also you notice that a lot of the work we do is for spaceflight missions. And so, yeah, that's understanding how the measurement would actually be made and what the constraints on that measurement are also incredibly important to our science.
Got it. So do you have like an integrated MS PhD? Because I already have a master's. So how much chance to get in?
Depends on the participating department. You'd have to look at it. I don't know if I can ask the ESS people. In astronomy, we do.
Okay.
And so maybe ESS planetary science would be a potentially better route for that. But we also offer a certificate. So if you sounds like you want to go beyond an MS, but for others who might want to just do an MS, do astrobiology, we also have a certificate program where, we can support people there as well. But you would have to try the participating departments to see whether, they would, I would try engineering for starters, to talk about that.
Okay. All right. Thank you.
I'm sorry, there's a gentleman at the back here who's had his hand up the entire time. I wonder if we can.
I'll get over there eventually.
Thank you. First, apologize for my poor English. It's not my first language. I'm kind of new to astrobiology, and you started with some questions that are leading the astrobiology. The main questions that you put there is, are we alone? What kind of life we can find outside? That made me think more in exobiology than astrobiology, because, well, exo, it's from the outside. And I'm wondering, now that we have more or better telescopes and devices to analyze exoplanets, and we have done a lot of work analyzing the living forms here on Earth, on extreme environments. Do we have any work trying to put these life forms on extreme environments in that exoplanets or that conditions to spread the life on Earth to the other planets? Because at this moment, I'm thinking that we are more focusing, trying to find life outside. But what about taking the life from here to the outside, not just analyze the plants in the space and that I mean, like, hey, what happens if we send some mushrooms to Mars? At this moment, we have, for example, the black mushrooms at Chernobyl that process all these radioactive things. So,
Right.
do we have any study on that?
Right. We generally try not to take things to Mars, I will say. So there's this whole planetary protection thing. I will say, though, that the BIOS endures thing I discussed at the end will be looking at one of the people in there is Sharon Doty, and Kevin Schaffman's here. Yep, Kevin's also working in this area. And this isn't answering your question head on, but it's an interesting aspect of this, which is, how do we grow plants outside of the Earth? How do we take life outside of the Earth and have it survive? Kevin works on this amazing thing called a holobiont, which is looking at how a plant needs microbes to do work for it, how there's an entire ecosystem. And so we understand you can't just take the plant out and plunk it down somewhere. I mean, people have tried to grow things in lunar soil, for example, but without all of this ecology of organisms that are required to keep the plant alive. And so there is some work now being done to try and figure out, okay, how do we grow these things in space? And that's not an extreme thing, but it is, a basic thing we need to survive. And yet, just trying to understand, okay, in that kind of an environment, which is a harsh environment for a plant, it doesn't have its soil and everything else it needs. So that work is certainly being done to try and push that there. Generally speaking, though, and I guess there's a treaty, I think. There's a treaty that says we shouldn't try and put life on other planets and other places just because of the preservation aspect of not, bringing in something that would contaminate that environment. So I don't know of any plans to do that, but I do hear all the time in the framework of colonizing the moon and colonizing Mars, of taking humans there to survive. We do think about plants and food, for example, but certainly, there's an entire division in NASA called planetary protection, which is specifically not to do that. But it's a very interesting question. And for exoplanets, of course, we don't actually understand enough about their environments yet. The very first exoplanet we could potentially look at to understand what its environment might be like is the TRAPPIST-1e planet. It could be a habitable planet, but we don't have the data on that one yet. So we don't know what environment we're dealing with there yet. But watch this space. Yeah, excellent question. Thank you.
Close to you.
Hi, my name is Michael. My background is in biology and computer science, and I saw that part of the presentation involved planets passing in front of stars, and I was wondering if potentially if there are any labs that might have data that's updated regularly, that's publicly accessible, so that you could potentially do a project involving the data from labs involving this field. Like, for example, like in terms of the medical field, there's some labs that have APIs where you can make dashboards of medical data as it comes out every day or week or whatever. Hopefully there's something like that in astrobiology. And also, I have a friend in the Netherlands, and it's 4 a.m. at 7 p.m. here, so they couldn't attend. I kind of also wonder if there's like if after the presentation, if the slideshow is available or something else in the presentation, so I could share with my friend who it's nighttime for them right now.
Excellent, excellent question. I can answer the last one first because that's easy. Yes, we recorded the whole thing. We will be putting it up on our website. So yeah, people who couldn't get to it because of the asynchronicity can watch it at any time. So there's that. Do we have data that you could analyze that would be relevant for exoplanet astrobiology? That's a toughie. I was going to ask Jim Davenport, Jake, anybody? Is Mario? Mario. Do we? Okay. I can translate through the microphone.
Okay, I'm gonna translate.
Okay, all right. So yeah, Jake is saying that on the NASA Exoplanet Archive, there is data that you can analyze yourself, and also sort of data from K2. And in fact, citizen scientists have discovered planets that way. So there is stuff you can play around with in the transit regime. Do those planets have life on it? Highly unlikely, but at least you'll get the thrill of discovering a planet. So that data is available to analyze if you're interested in that. We need more than one microphone runner.
We need a microphone runner that's not in heels.
You can always take them off.
Thank you. So I am an undergraduate, and I guess I was wondering if for people without that strong of an academic background yet, how they can be involved in astrobiology, not just the department, but the science in general, and to learn more with events such as these, because from a newcomer perspective, astrobiology seems pretty intimidating and like a specialized kind of field. So I guess it's just me asking about accessibility for folks who are kind of new.
In undergrad, yeah, sure. I mean, we do have projects often in the summer. I mean, you can start by asking the astrobiology faculty if they have any REU opportunities for the summer. So we bring in undergraduates from all sorts of different backgrounds to do projects like that. So you could maybe start with that. I'd also encourage you to maybe listen in on some of our colloquia, learn a little bit more about the field that way. You go through the YouTube channel and see if there's things there that interest you in particular, so you can sort of get a better idea of what you're interested in, maybe before asking someone about whether they have research projects in that area. But it's certainly, I mean, we all start somewhere. I know the field maybe seems intimidating, but I don't think it's any more intimidating than science itself. And in that case, as I've told my students on workshops, it's often a lot less intimidating than you'd think. And so, yeah, if you come in for an REU or something like that, people will train you on the tools that you need and to get you up to speed and everything. We won't be expected that you will know all that stuff in advance. That's the whole point of learning in a research experience. Yeah. This guy who's had his hand up for forever.
You gotta turn it on.
You gotta turn it on.
It's easy.
Hi,
There's a button somewhere.
sorry, I'm online. Hi, my name is Anjali.
on top.
I'm currently
It says
working.
on/off.
See what I mean?
Hi, my name is Marcus. Thank you so much. This was fantastic. Of all things, I went to UW and I took an astrobiology course when the book Rare Earth was first published. So this is kind of wild. But I guess what I'm curious is, given how diverse your field is, where do you think are the areas where the next insights or discoveries might come from? Or what are the sort of hotly debated or where are the sort of nuggets of interest in your field these days?
Oh,
Thank you.
wow. And you asked me to do that in front of the entire program. Okay, so not your stuff. Yeah, so I think, well, I mean, okay, I'll be biased and say that, exoplanets, we are, on the cusp of being able to get data on a potentially habitable exoplanet, and that's TRAPPIST-1e. And so we'll all be looking, I mean, does it have an atmosphere? It may be a completely blasted bare rock, right? So these are sort of really big questions about whether we can have habitable environments out there. And I mean, and I think, honestly, data is coming down on that already. And the question is whether we can analyze it because of the noise from the star. But yeah, so I think that's coming up in the next year. That's probably going to be something that comes out that is just super, super interesting. I am very interested in the habitability of M dwarfs overall because of this problem. I called out the fact that they might have all of their atmospheres and ocean blasted off early on. But then what happens to a planet after that? The star then dims. It keeps outgassing. It's not the end. What happens to that planet? Can it recover an atmosphere? Can it recover an ocean? What is the long-term habitability of these types of planets after a particularly violent early career? And so, yeah, I'm just interested in the interaction of planet and star, how that affects habitability and how things recover, and what sort of the likelihood is that the planets that we will be able to look at in the next 10 to 20 years are actually able to be habitable. So I will speak from my field. Are there others in other fields? And there's a lot of faculty here who'd like to maybe say, what's the cutting edge of your field right now? What's things you want to learn about? I know Woody wants to find life that transmits to us, so he's a big SETI proponent. Yeah, Hillary. Okay, Hillary's going to go for it. So Hillary is the chair of oceanography.
So I am waiting with bated breath for Europa Clipper data to test hypotheses that we've set from lab stuff.
Okay, yes. So we have missions going to other habitable environments potentially in the solar system. So yeah. So Europa Clipper should hopefully tell us whether we have a subsurface ocean and whether or not it is habitable or able to, or has life in it. At least habitable. Anybody else? Oh, there's someone over here and there's someone back there. Okay, Mike first and then next.
Do we have an understanding of comets or asteroids from other solar systems that offer opportunities to explore that are different than exploring star systems using remote sensing or visiting planets in our own solar system?
Yeah, that's a really interesting point, because sometimes, stuff ends up in our solar system that came from somewhere else. So, I mean, the first famous one of that, of course, was the Oumuamua object. They do tend to come in at really high speed, though, and then leave very quickly. So I'm not sure whether anybody's planning any sort of sample intercept return or whatever from that type of an object, but I would imagine that would be a very high scientific priority for exactly the reason you stated, is that it would tell us something about the makeup and potentially the evolution of another planetary system other than our own. So yeah, does anybody here in the engineering department? Oh, Mario, do you know? You're getting an all-star faculty cast here.
There's a European mission called Comet Interceptor that originally was built to go and chase a comet in our solar system, and then interstellar objects were discovered. So they're going to launch something that will essentially hover around the Earth and wait for an interesting object to show up and then go after it. If we discover with this other mission that UW is involved in called Rubin Observatory, we discover a comet that has on the right trajectory, is slow enough to get caught, then that's where that mission is going to go. So next 10 years could be exciting.
Great. Question. We should also get some questions from our virtual audience after. Do you have anything from virtual? No? Cool. Okay.
I'm an undergrad in ecology right now, but a lot of my work has been in science education. I'm an educator at the Pacific Science Center. And I was wondering, I had two questions kind of related to science communication. I was wondering, as like an astrobiologist, what do you want like the public to know about astrobiology, or like a common misconception you want dispelled? And I was also wondering, as an astrobiologist, like what your favorite sci-fi movie or book is.
Okay. Arrival for the last one.
So good. That's one of my favorites.
That's
Thank
your
you.
favorite too. I mean, it's just brilliantly done. And this whole concept of that communication, trying to communicate. That's where I love Arrival. I think one of the biggest misconceptions in astrobiology, and I realize I didn't dispel it at the beginning, is that a lot of people think we're looking for aliens, like humanoid aliens, right? Whereas astrobiology is really about trying to find probably microbial life. I mean, if you look at the life on our planet, the microbes have had the biggest impact on our environments, on our atmosphere, and over time, you know, the oxygen you're breathing right now, care of cyanobacteria, right? And so I think a lot of people think that when we're doing astrobiology, we're hunting for little green men, and that's why they say, hey, you're a science in search of a subject. You don't have your alien. But what we're really trying to do is understand ecosystems where life, even if it's microbial life, is present. It can't build a radio telescope, it can't talk to us, but it can change its environment, and it can tell us that it's there by doing that. So that's really what we're trying to do. And I think the argument there, if you understand the Drake equation, is that, you know, we'll get to intelligent life maybe as a low probability outcome at the very end, but the higher probability outcome is that we do develop life within an environment. You can't build radio telescopes, but it's still there, and we want to learn about it. And that's a lot of what astrobiologists are focusing on.
You should read Project Hail Mary by Andy Weir. So good. Very good.
Okay.
Continue.
Anybody else want any recommendations for science fiction movies or books?
Other questions? Or are there Zoom questions?
There's maybe one.
Sure. Are there any past or current research on synthetic life at UWAB?
Oh, it's a shame John isn't still here, is he? Roy, is Roy here? Roy would be the closest thing.
I don't think he's here.
No, okay, Roy's not here either. Okay. Lauren, do you know? No, I don't think so. Yeah, so Dave, well, I mean, some people have been trying to recreate protocells and things like that. So in Sarah Keller's lab is kind of the closest thing we have to that. But there is a scientist, Dave Deemer, who has worked on trying to generate, you know, sort of cells in the lab and sort of work on synthetic biology, but he does not work here. But yeah, but it is a field, but nobody here, as far as we know.
I'm coming. No, wait, Mike's coming. Mike's got it.
Yeah, Mike's got it.
Probably have about 5 to 10 more
Sure.
minutes.
I've been standing up for a long time.
Awesome. Thank you so much for this. This has been so interesting. I don't have an illustrious degree like the rest of you in the room. My background is in journalism, which
we
means
need
I'm
that too.
very human focused and story focused. So I'm curious what your perspective is on. Do you need this one?
When or if? Yes, go ahead and project.
Repeat the question.
Yes, I will repeat that. So we've talked a lot about trying to find life elsewhere. But the question from a journalist is, you know, what is our perspective of what science will do next once that discovery is, I guess, reported? Have I missed something there? Was there a social aspect? No, that's good. Okay, perfect. I'm so glad you asked that because that was one of the slides I skipped over. So, so basically, we have thought about this a lot, and there have been NASA workshops as well on communicating the discovery of life in the universe, things to think about. I think one of the things as scientists we want to do is we want to make sure we're as confident as possible. So if you actually do a timeline of how many claims of life or there have been in the last decade or so, it's actually increasing with time. And so we're seeing claims of potential life detection coming out, for example, you know, phosphine on Venus, or there was some sort of microbe, I can't remember the Zeo thing. So there's a bunch of things that have been coming out, and we as scientists are starting to grapple with this, that that claims of life detection are not just theoretical anymore. They are happening. We need to deal with them. And so we as scientists actually came together in a community workshop called Standards of Evidence, for bisignature claims, essentially. And so what generally happens, and the one I'm most familiar with is the phosphine on Venus one, because we actually leapt in and Andrew led a paper trying to figure out what was going on. So what happens is someone comes out with a claim that, we've discovered phosphine on Venus and it could be a sign of life. Right? That's an amazing claim. We'd love it to be correct. But on the other hand, we also don't want that to keep going if it's not correct. And so what happens is, and this happened organically and spontaneously, which is why we were trying to maybe come up with a formula for doing it. But first of all, scientists will go and check the data. Okay, we're all big about replication. We generally don't replicate anything unless it's this big a claim, right? And when it's this big a claim, scientists will come out and they will start trying to reanalyze the data, confirm the detection, you do all that kind of thing. Do we have a real detection? Have we maybe mislabeled it in some way? That sort of thing. Those are the first two things that we will check. After that, if there's a claim of life detection, it looks like we've got the right measurement, we have the right molecule, we'll ask ourselves, okay, is this something that the planet could have created without life? And so a bunch of people will go off and do some modeling or laboratory work on that. And then the second question is, does it make sense that life would produce this signal in this environment, right? And in the Venus case, for example, they were saying there was phosphorus actually above the clouds, but phosphine only has a lifetime of several seconds above the clouds. And so the amount of stuff that had to be pumped out was something like 10 times the sort of productivity of our own photosynthetic biosphere. So it didn't make a lot of sense that life would be capable of producing that kind of signal in that environment. And then finally, we'll be trying to figure out between the life and the non-life hypothesis. There'll be other questions we'll want to ask and other measurements we want to make to try and see if we can, close the deal on one or the other hypothesis to say, yeah, okay, this seems to be probabilistically much more likely one. So I think what you will see as these claims come out is that the community, will get together, and the observers in this case, the people who had used ARMA before, went and checked the data. Other people tried to do the modeling. People who analyze spectra and do retrieval or radiative transfer tried to fit the spectrum and tried to interpret it. So yeah, I think we're not going to stand around and just accept it. There will be work that will be done. And, the point is not to be mean. The point is to, can we confirm this? If we can, it's awesome, right? And if we can confirm it, then there's questions about, whether or not it's valid, because we don't want that to stand. And I will say that when I'm talking about getting rid of misconceptions, one of the misconceptions we were told in this workshop was that most of the community thinks we've discovered life already. I mean, there've been enough stories about, things happening that what we'll have to do is actually convince people that no, we haven't actually discovered life already. This is the first one. So yeah, and that's kind of challenging things to think about. But NASA is funding workshops on this right now to think about what we would do and what the protocol would be if these claims come out.
Come.
I was curious about the difference between 3 and 5. Like I see 3 as abiotic sources for detection, and then I see 5 as independent lines of evidence to support a biological or non-biological. So I understand from my biology degree that abiotic would be a synonym of non-biological, but the difference between 3 and 5 would be, I wish that I did that.
So the idea is that when you get this signal, you've confirmed it's real, you haven't mistaken it for something else. There's 2 lines of inquiry. One is, can I produce this abiologically without biology? And does it make sense that life produces this in this environment? And so we'll explore those lines. But as we develop this, question 5 is really like, okay, what's the next experiment to distinguish between them? So in some ways it could be the same as 3, but it's just once we've done this initial study, is there a single piece of evidence that would help us decide conclusively between this hypothesis or this hypothesis? And question 5 then goes and gets that information. Does that make sense? Yeah, okay.
We have time for maybe one more question.
It's a good question.
Mike, does your mic work? Mike, does your mic work?
I guess I'm sort of curious, and this might be a little bit far-fetched, whether or not there's any research done on quote-unquote non-carbon life forms within the community.
Yes. So that comes up all the time. So okay, there's a number of ways of addressing that. We do tend to work more with water, using water as a solvent, carbon-based life. It has a lot of chemical advantages, and so people have, so for example, silicon-based life is often posited, but the interactions and the reactions that have to occur there tend to be very slow. They're not very varied. It's just not a good substrate for the kind of versatility and the bonding and breaking that we need for life reactions to occur at a reasonable time and space. So there are some really, if you want to follow up on this, there's some really great papers. So there's one by Pohorille and Pratt, Pohorille and Pratt. And they look at water as a solvent for life and talk about just all of its chemical advantages and how it is, in fact, well suited to be a solvent for life. So people also talk about, what about other solvents like ethane or whatever? Yes, and we have John here too. John, would you like to talk on this as well? No. Yes, oh yes, the Weird Life Report would be a good one to look for too. What is that called again? I don't know what it's called, but yes. Was there what, sorry? The distribution of organics in the solar system. Okay, so yeah, so that National Academy of Sciences, if you go search on their website, the it's not called the Weird Life Report. What's it called? Is distribution of organics? Is that what it's called?
I think it is the distribution of organics.
Anyway, go search for that. You can probably put some keywords in and find it. But there are, I mean, people, initially we were a little bit parochial and we were just looking at carbon-based life in a water-based solvent. But there has been a lot of work done in the last couple of decades to actually show that that makes a lot of chemical sense. And so you can read some of the chemistry that is involved and why it's actually a really good solvent for life, for the sorts of things we're looking for. That's not to say there can't be other forms of life out there, but certainly, and I always remember a quote from Jim Green, who was the head of Science Mission Directorate at NASA, and he said, the other thing about water is it's the most common polyatomic molecule in the universe, and the solar system is squelching with it. It is everywhere, right? So that's another thing, just the ubiquity of the solvent, the fact that it's liquid over a very high temperature range, all that sort of stuff really points us towards not just using Earth-based carbon-based life because we're lazy, but because it actually makes a whole heck of a lot of sense chemically for the kinds of reactions that we need. So people have done some work on it. That's not to say we're not biased, but at least we've backed that bias up with science. Oh yeah. Yes. Okay. So the other type of biosignature, so I talked just very briefly about oxygen as a biosignature, but we are now expanding the field. This is the frontiers of the field. We're now expanding the field into what we call agnostic biosignatures. So great. I have this teleprompting in the front here. So agnostic biosignatures are really our chance at getting at life that we don't know, life as we don't know it. And so in what we call a targeted biosignature, we hypothesize a metabolism and we say, well, this metabolism would use its environment in this way and it would produce this product. And so we go look for that product or this thing being used in this way. But for agnostic biosignature, what we're looking for is something anomalous in the environment that we can't explain with a planetary process. So my best description of what an agnostic biosignature is it's not planet. Okay, we're looking for a signal that is not planet. And so the easiest one I can describe to you is if you're looking at an extrasolar planet and you see two gases out of equilibrium. What that means is if you left those two gases to their own devices, they would destroy each other and they wouldn't be there. So the fact that they're there means that there has to be a flux of them from somewhere replacing them as they get destroyed. And if that flux is high enough, then you get, a steady state of that gas in the atmosphere. And so the biosignature is not the disequilibrium, it's the fluxes that are maintaining the disequilibrium against the destruction. And those fluxes could be, I mean, maybe there's a flux of neon on a planet and we think, well, there's no geological or other process that we can think of that would just have neon coming off the surface of the planet, for example. That's a silly example, but you know, something where it's just not, there's just no known planetary process. And the thing about agnostic biosignatures is, yes, it breaks us out of looking for a specific metabolism, methanogenesis or photosynthesis or whatever. But at the same time, you have to understand the environment you're looking at exquisitely well to be able to tell that what's going on is not due to the environment. So there are pros and cons in doing this. Either you study your environment to death so that you know that this perturbation is not due to a planetary process, or you go after targeted metabolism because you know it's something that life does in this particular environment. But I think agnostic biosignature is exciting because, yeah, it could allow us to hone in on processes that don't make any sense and that may ultimately be due to some form of alien life. Right, okay, sorry. You. So Mary, I mean, yeah, so I mean, I used to call it, I mean, not just looking for not life, but I used to say that you know, an agnostic biosignature was really just like a disturbance in the force. It was something that didn't make any sense, you know, in its environment, that something that was off. And so that's what we'll be looking for. We'll be looking for, you know, when we get a good spectrum of an extrasolar planet, we'll be looking for something that doesn't make any sense. And we will go after that. It's either a new planetary process or potentially it's life.
All right, let's all thank Dr. Meadows again for the talk. I have a total of 2 sentences to say, I promise. I'd also just like to thank